Welcome to Studious. Today we begin an entirely new series, one that takes us into the world that exists beyond the resolution of the human eye. Microbiology is the scientific discipline that studies microorganisms, life forms so small they require the power of microscopic magnification to be observed and characterized. Yet despite their invisibility to the naked eye, these organisms govern virtually every dimension of life on Earth. They recycle nutrients in the soil, drive oxygen production in the oceans, regulate the human immune system from birth, cause the most lethal infectious diseases in history, and are now being harnessed to manufacture life-saving pharmaceuticals. This introductory video lays the conceptual foundation of the entire field, from its etymological roots and defining parameters, through its historical pioneers, to a complete structural taxonomy of its core branches and applied fields. Whether you are a first-year paramedical student, preparing for USMLE, or simply beginning your journey into biomedical science, this session will give you the mental map you need to navigate every subsequent topic in microbiology with clarity and confidence.
Before we study organisms, we study the name itself, because understanding the etymology of a scientific discipline immediately encodes its entire scope. The word microbiology is constructed from three Greek roots. The first component, micro, derives from the Greek mikros, meaning small, specifically small enough to be invisible to the unaided human eye. The second component, bio, from the Greek bios, means life, referring to living organisms. The third component, logi, from the Greek logos, means study or knowledge. Combined, microbiology therefore means the study of small living organisms. That is, the study of microbes that cannot be visualized without the assistance of a microscope. This seemingly simple definition contains an important implication. Microbiology is a discipline defined not by phylogenetic relationship or evolutionary lineage, but by scale. Any living organism that falls below the threshold of naked eye visibility is a legitimate subject of microbiological inquiry, regardless of whether it is a bacterium, fungus, protozoan, alga, or even a microscopic metazoan. This scale-based definition is what gives microbiology its extraordinarily broad scope across biology, medicine, ecology, and industry.
Having established the linguistic framework, let us now precisely define what qualifies as a microorganism. Three parameters are used to characterize microbial life. The first is the scale threshold. Microbes are generally defined as organisms measuring less than 1 millimeter in size. It is worth noting that this threshold is a guideline rather than an absolute boundary. Recent scientific discoveries have identified rare anomalous organisms, such as Phytomargarita namibiensis, a sulfur-oxidizing bacterium discovered in Namibian sea sediments, which can grow to nearly 1 centimeter in diameter, technically visible to the naked eye, yet unambiguously classified as a microorganism by its biology and lifestyle. The second parameter is cellular complexity. Microbes may be unicellular, consisting of a single autonomous cell, as in most bacteria and protozoa, or multicellular, as in certain fungi that form complex hyphal networks. The third parameter is cellular structure. Organisms may be prokaryotic, lacking a membrane-bound nucleus, as in bacteria and archaea, or eukaryotic, possessing a defined nucleus enclosed within a nuclear membrane, as in fungi, protozoa, and algae. These three parameters together define the enormous biological diversity encompassed within the single term microorganism.
Every scientific discipline can be traced to transformative individuals whose insight redirected the trajectory of human knowledge. Microbiology belongs to two such figures. The first is Antony van Leeuwenhoek, a 17th century Dutch cloth merchant turned self-taught lens grinder from Delft, Netherlands, who is universally recognized as the father of microbiology. Between 1674 and 1683, using single-lens microscopes of his own construction that achieved magnifications far beyond anything available at the time, Leeuwenhoek became the first human to directly observe and describe living microorganisms, including bacteria from dental scrapings and protozoa from pond water, which he called animalcules. His meticulous observations, communicated in letters to the Royal Society of London, opened an entirely new dimension of biological reality. The second giant is Louis Pasteur, the 19th century French chemist and microbiologist recognized as the father of modern microbiology. Pasteur dismantled the prevailing doctrine of spontaneous generation through his elegant swan neck flask experiments, established the germ theory of disease, demonstrating that specific microorganisms cause specific infections, developed the principle of pasteurization, and pioneered modern vaccination. Together, Leeuwenhoek's discovery and Pasteur's mechanistic framework built the entire edifice of modern microbiology.
Microbiology is not a monolithic discipline. It is a vast intellectual territory organized into two major divisions. The first division encompasses the core branches, which are the pure scientific arms of the field, concerned with understanding microorganisms at a fundamental level. These core branches are further subdivided into two categories: organism taxonomy branches, which are defined by the type of organism being studied: bacteria, fungi, protozoa, algae, parasites, and systems and processes branches, which are defined by the functional or mechanistic aspect under investigation, such as genetics, physiology, ecology, or morphology. The second major division encompasses the applied fields, areas of microbiology oriented toward practical outcomes in human health, food production, agriculture, industry, and emerging scientific frontiers. Applied microbiology does not merely observe microorganisms. It deploys, controls, or combats them to achieve specific objectives. This two-part taxonomy, core branches on one side and applied fields on the other, is the organizing framework around which all of our subsequent discussions in this series will be built. Understanding it now will give you an intellectual map that makes every future topic instantly locatable within the broader structure of the discipline.
The first category of core branches is defined by organism type. Each subdiscipline is dedicated to a specific taxonomic group of microorganisms. Bacteriology is the study of bacteria, prokaryotic, single-celled organisms without a nucleus that represent the most numerous and metabolically diverse life forms on Earth. Bacteria include both beneficial organisms, essential for human gut health and nutrient cycling, and dangerous pathogens, responsible for diseases such as tuberculosis, cholera, and pneumonia. Mycology is the study of fungi, including yeasts, molds, and mushrooms, eukaryotic organisms that can be unicellular or multicellular and are critical in both ecology and medicine, causing infections such as candidiasis and aspergillosis. Protozoology is the study of protozoa, single-celled eukaryotic organisms with animal-like characteristics, notorious for causing devastating parasitic diseases, including malaria, caused by Plasmodium species, and sleeping sickness, caused by trypanosoma. Algology, also called phycology, is the study of algae, photosynthetic eukaryotic microorganisms that produce approximately 50% of global atmospheric oxygen and underpin the base of aquatic food chains. Finally, parasitology is the study of parasites, organisms that live in or on a host at the host's expense, including protozoa, helminths, and ectoparasites, many of which carry enormous global disease burden.
Beyond organism-centered taxonomy, a second category of core branches approaches microbiology through the lens of biological systems and functional processes. Microbial ecology examines the relationships and interactions between microorganisms and their environment, including the composition of microbial communities in soil, ocean, human gut, and extreme habitats such as hydrothermal vents. The human gut microbiome, now recognized as a critical regulator of immunity, metabolism, and even neurological function, is one of the most active areas within microbial ecology today. Microbial morphology concerns itself with the detailed physical structure and shape of microbial species, cell wall architecture, flagellar arrangement, spore formation, characteristics that form the foundation of microbiological identification and are routinely used in clinical diagnostic laboratories. Microbial physiology investigates how organisms survive, acquire nutrients, generate energy, and manage their metabolic networks, including how certain bacteria survive extreme heat, acidity, or radiation. Microbial genetics studies the genetic material contained within microbial cells, chromosomal DNA, plasmids, and mobile genetic elements, including the mechanisms of antibiotic resistance gene transfer that underpin one of medicine's greatest contemporary challenges. Finally, molecular microbiology focuses on the internal biochemical reactions driving cellular metabolism, replication, transcription, and growth at the molecular level.
The applied fields of microbiology are where the discipline directly intersects with clinical practice, public health policy, and the life sciences industry. The first and most clinically central applied field is medical microbiology, the branch dedicated to identifying the causative microbial agents of infectious disease and developing the diagnostic procedures used in clinical laboratories to confirm infection and guide treatment. Medical microbiologists characterized SARS-CoV-2 as the causative agent of COVID-19, identified Mycobacterium tuberculosis as the cause of tuberculosis, and continue to profile emerging pathogens. Public health microbiology extends this mission from the individual patient to entire populations, monitoring the spread of infectious disease across communities and implementing societal control measures, from vaccine campaigns to quarantine protocols to the epidemiological modeling that drove COVID-19 lockdown decisions globally. Immunology and serology form the third pillar of this applied health cluster, investigating the body's defense architecture, the innate and adaptive immune responses, antigen-antibody interactions, complement activation, and serological diagnostic tests that detect exposure to specific pathogens based on antibody presence in patient serum. Together, these three fields form an integrated defense network that underlies modern infectious disease medicine.
Microorganisms are not only agents of disease, they are also powerful tools for food production, agricultural sustainability, and consumer protection. Agricultural microbiology harnesses microbes to enhance crop growth, protect plant health, and increase agricultural yield. Nitrogen-fixing bacteria, such as Rhizobium species, form symbiotic relationships with legume roots, converting atmospheric nitrogen into bioavailable ammonia and enriching soil fertility without chemical fertilizers. Mycorrhizal fungi extend root systems, dramatically increasing water and mineral uptake. Microbial biocontrol agents are increasingly used as alternatives to chemical pesticides, representing a major frontier in sustainable agriculture. Dairy microbiology leverages the biochemical activities of specific microbial strains to drive fermentation-based food production. The most familiar example is the use of lactobacillus species, lactic acid bacteria that ferment the lactose in milk into lactic acid, curdling milk proteins and producing yogurt, curd, cheese, and ghee. These microbe-food partnerships are thousands of years old and remain commercially vital. Food microbiology takes a protective orientation, studying microbial contamination in the food supply, characterizing spoilage organisms, investigating foodborne pathogens such as Salmonella, Listeria, and E. coli 0157, and developing preservation technologies including pasteurization, refrigeration, modified atmosphere packaging, and antimicrobial surface treatments to extend shelf life and protect consumers.
One of the most economically significant applications of microbiology lies in industrial bioproduction, the large-scale use of microbial metabolic machinery to manufacture commercially valuable compounds in industrial fermenters. The principle is elegant. Microorganisms are extraordinarily efficient chemical factories. Given appropriate nutrients, temperature, and oxygen conditions inside a bioreactor, they can convert raw inputs into high-value outputs with remarkable specificity and scale. The alcoholic beverage industry depends entirely on Saccharomyces cerevisiae, brewer's yeast, to ferment sugars from grains, grapes, or fruits into ethanol, producing beer, wine, and spirits through controlled anaerobic fermentation. The pharmaceutical and nutraceutical industries exploit microbial biosynthesis for vitamin production, including vitamin B12, which is exclusively produced by microorganisms and cannot be synthesized by animals or plants, and essential amino acids used in dietary supplements. Most dramatically, the antibiotic revolution was built on microbial production. Alexander Fleming's discovery of penicillin from the penicillium mold in 1928, followed by the industrial-scale fermentation of penicillin developed during World War II, established the template for microbial pharmaceutical manufacturing. Today, bacteria and fungi produce not only antibiotics, but also hormones such as insulin, first bioengineered in Escherichia coli in 1982, monoclonal antibodies, vaccines, and a growing armory of biologic drugs that define modern medicine.
The outermost applied frontiers of microbiology push the discipline beyond the terrestrial and into entirely new environments. Aquatic microbiology studies the microorganisms inhabiting marine and freshwater ecosystems, from the phytoplankton communities of sunlit surface waters to the chemolithotrophic bacteria of deep-sea hydrothermal vents that sustain entire ecosystems without sunlight, and to the microbial biofilms that colonize drinking water distribution systems and pose persistent public health challenges. Aeromicrobiology, or air microbiology, investigates the community of airborne microorganisms and aerospora, fungal spores, bacterial cells, and viral particles suspended in the atmosphere that are responsible for spoiling stored food, triggering allergic disease, transmitting respiratory infections, and damaging agricultural crops through windborne pathogen dispersal. The most visionary of the expanding frontiers is exomicrobiology, the study of microorganisms capable of surviving and thriving in the extreme conditions of outer space. Research aboard the International Space Station has demonstrated that certain extremophilic bacteria, such as Deinococcus radiodurans, can survive intense radiation, desiccation, and the vacuum of space. Extremophiles found in Earth's most hostile environments, boiling volcanic springs, hypersaline lakes, and Antarctic permafrost, provide model organisms for astrobiology, offering tantalizing clues about the possibility of microbial life on Mars, Europa, or Enceladus.
This final slide captures everything we have covered today in a single, elegant framework, the micro-macro spectrum of microbiology. At the center sits the microbe itself, an organism smaller than 1 millimeter, invisible to the naked eye, yet the organizing principle around which this entire discipline revolves. Radiating outward from this central point are 4 concentric rings of influence. Ring 1 is the human body, where microbiology manifests as immunology, diagnostic medicine, infectious disease, and the microbiome. Ring 2 is global industry, where microbiology drives food production, dairy fermentation, pharmaceutical manufacturing, and the biotechnology sector. Ring 3 is the planet, encompassing microbial ecology, ocean biogeochemistry, soil health, agricultural microbiology, aeromicrobiology, and aquatic systems. Ring 4 is the cosmos, where exomicrobiology pushes the boundaries of life science to zero-gravity environments and the possibility of extraterrestrial microbiology. From entities smaller than a millimeter to the vastness of outer space, the invisible world truly governs the visible one. This is the scope of microbiology. This is what we will explore together in this series. If you found this video valuable, please hit the like button. Drop a comment below with any questions or topics you want covered next. Share this video with a fellow student and subscribe to Studious and hit the bell so you never miss a new episode. Let's keep studying together.